All right, class, welcome back. So today we will be doing 7.4.1 study on DNA fingerprinting. There's 15 pages to it, and I will be filling out the study guide, and you can follow along. I will be reading the study, so you can follow along there if you'd like. What I will do now is get started. I'm sure everybody's very excited that this is the last study guide on DNA, which can be kind of a tough subject, especially trying to teach it to yourself from home. So let's get started. Page one. You and your friends may like the same music, have the same hobbies, and share opinions on a variety of topics, but how similar is your DNA? So it says, how much of your DNA do you think is the same as your friends? Well, you know it's not 100% because if you shared 100% of DNA, then they would be just like you, 100% just like you. But it's pretty darn close to 100%. I'm going to say E, 99.9%. Yep, correct. The DNA of two humans is mostly the same. I'm going to go to the next page. Every human's DNA is 99.9% the same as every other human's. You and your friend's DNA are, flip it on airplane mode real quick, that way we don't have any disruptions. You and your friends are incredibly similar genetically speaking. 99.9% of your DNA or your genome is the same as theirs. So we'll put that down. How similar is the DNA of any two humans? 99.9%. Very similar. But wait a second. If your DNA is so similar, how is it you and your friends don't look exactly the same? The 0.1% difference may seem small, but it represents about 3 billion pairs of nucleotides. And that's a pretty big difference, right? It's just that there's so many base pairs that even a small percentage is a big number of base pairs. Those 3 billion base pairs can explain all the differences in people's traits, such as skin, color, height, and hair texture. So the DNA difference among people is useful to scientists studying the human genome. The differences can also be useful to society. Can you guess why? So you can fill in down there in 40 words or fewer. Why do you think that 0.1% difference in DNA could be useful to society? So think about like when you hear about DNA in the news. So fill that in and hit submit, and I'm going to go to the next page. So you can pause it to fill that in. And when you come back, we'll be on page three. All right, page three. DNA fingerprinting is useful at crime scenes in tracing family history and in medicine. So we have this technology called DNA fingerprinting, and it's used to look at unique patterns in a person's DNA. In forensic science, DNA fingerprinting can help identify the victim at a crime scene, identify a person who has committed a crime, or clear the name of an innocent person who was accused of a crime. DNA fingerprinting can also help determine individuals' family heritage, including who their parents are, as well as their ethnicity or where their ancestors come from. You might have seen this on an episode of Maury. Are you the father? Are you not the father? Right? So that's DNA tests. In medicine, DNA fingerprinting is used more and more commonly to help provide medical treatment that is as specific as possible to a patient's needs. One important use of DNA fingerprinting is in medicine, is to identify a good match for organ donation. So here's a little opinion piece down here. What part of DNA fingerprinting sounds most interesting to you? So you can click on one of those, forensic science, ethnicity, family research, and medical treatment, and we will go on to the next page. Uh, and actually, right before we do that, let's go in and fill in uses of DNA fingerprinting. All right. So, how is DNA fingerprinting used in forensics? It can identify victims and suspects. All right. So that, and it can help figure out who committed a crime. All right, so they always talk about finding DNA in places and being able to match those to people that might be accused. It could even be used to clear people. That was not your DNA. So how is it used in medicine? So it's used to identify treatments for a particular person. And how is DNA fingerprinting used to help people learn about their families? So you can learn about your ethnicity. So maybe what country, if you live here in the United States, what country maybe your ancestors came from. And you can figure out the parents of the child. So parentage, you could say your heritage. These are all things that you can kind of figure out. All right, so let's go to page number 4. So DNA samples come from a variety of sources. The first step in DNA fingerprinting is getting a DNA sample. What do you think could be used as a DNA sample? So you can type in a few words there and submit it and figure out where do you think that a DNA sample could come from. All right, so you can put that in there. You can pause it, and I'm going to write in here where I think that things that you could get DNA from. So, all right. So let's go to page number 5. All right. So page number 5 is an exercise to help us review what we've learned so far. So click through that and see how you do. And it'll let you know if you get any answers wrong. And then you can pause this video and you can click through that. And then when you're finished clicking through, you can restart this video and we will be on page number 6. All right, so once we have a DNA sample, we need to make more DNA because maybe there's not enough DNA to test. So we use a process called PCR, which stands for polymerase chain reaction. All right, so polymerase chain reaction is a process where we can take a small amount of DNA and we can make a larger amount of DNA from it. So we see here in this picture, it says PCR is done many times, doubling the number of copies every single cycle. So it lets you fill in here in 25 words or fewer, predict how many strands will be in the 5th cycle. So we have the first cycle has 2, the second cycle has 4, the third cycle has 8, looks like the 4th cycle has 16. So how many do you think are going to be in the 5th cycle? You can fill that in, and I will explain what PCR does for our step 2 of our study guide. So it makes many copies of a sample of DNA. Perfect. All right, so let's go to page 7. We're going to learn about restriction enzymes. So after DNA, the sample has been made larger using PCR, scientists use things called restriction enzymes to cut the DNA into fragments or bands of varying sizes. The fragments of each person's DNA are unique, allowing scientists to make a DNA fingerprint that can identify an individual. Each restriction enzyme recognizes a specific region of DNA and cuts the DNA up at that point. Answer the questions to learn more about restriction enzymes. So we have this picture down here, right? And it's a strip of DNA, and it says that there's 3 cut sites in it, 1, 2, and 3. All right. So if the restriction enzyme has these three sites where it makes cuts, how many pieces of DNA would be left? All right. So we have one piece that's being cut one, two, three times. And if you count, you'd have one, two, three, four pieces of DNA that are left if you cut the one piece three times. It says imagine a strip of paper that you cut three times. You'll have four pieces of paper. All right. And the next part, you're going to fill in your answer here. It says, how does the use of different restriction enzymes affect the size of the DNA bands produced? So we have three different restriction enzymes being used, A, B, and C. And what BP stands for is base pairs. So you can see that these three different enzymes actually cut it. They cut the strip. They cut the strip of DNA in different spots. So by using different restriction enzymes, you're going to end up with different size fragments of DNA. So number three, what does restriction enzymes do? So they cut DNA at specific sites. All right, let's go down to page number 8. Gel electrophoresis. So big word for a process that we use in DNA to study DNA. So once the DNA has been cut up by the restriction enzymes, samples can be analyzed using gel electrophoresis. Samples are loaded into little wells in a gelatin-like material. Then the electricity is used to pull the DNA samples through the gel. Electricity can move the DNA because the DNA molecules naturally have a negative charge. On the left, you see a scientist putting a DNA sample into a gel. On the right, you can see the gel hooked up to a source of electricity. Each portion of the DNA in the sample will travel a certain distance through the gel, and that distance depends on its mass. So what does gel electrophoresis do? So it separates the DNA by mass. Separates the DNA fragments by mass. We will go to page number 9. All right. A review of terms. So you can go through here and do your review of terms, try to match them up. And you can pause this video. And when you're finished, you can start, and we'll be on page number 10. All right, so we're on page number 10. Looking at a DNA fingerprint. A DNA fingerprint is an image of a gel electrophoresis result. After gel electrophoresis, imaging techniques are used to preserve a picture of all the DNA fragments. The images allow fragments from different samples to be compared. So here we have a picture of the gel electrophoresis, and each sample is read from left to right. You can see the lines, and those lines represent different pieces of DNA and where they ended up when they slid across the gel. So it says during the process of gel electrophoresis, fragments of negatively charged DNA are placed into wells on the gel bed, and then the fragments of DNA are then pulled towards the positive side. Why do some of the DNA fragments travel farther than others? So we talked about how some of the fragments travel farther because they have less mass. They are smaller. So the smaller fragments are able to move faster through the gel, and that's why they end up further to the right. All right, so our next, it says click on the link at the right to see a DNA fingerprint. Each small line represents a fragment of DNA, and each set of lines is called a lane. All right, so we'll click on that. See the DNA fingerprint. Okay, so we got these blurry lines. All right, So let's look at this. It says, which two lanes appear to have the same pattern of DNA fragments? 1 and 6. So let's look at 1 and 6. 1 and 6. No, 1 and 6 aren't the same. They start to look the same at the beginning, but you start to go down a little bit. They're not quite the same. Let's look at 3 and 10. 3 and 10. 3 and 10 look pretty close to each other. Let's keep going. Make sure 5 and 11. 5 and 11. Not quite right. Those blue ones on the top don't line up. 6 and 12. 6 and 12. I'm going to say, yeah, 6 and 12 don't line up by the looks of it. I'm going to say 3 and 10. Yep, the two lanes appear to have the same pattern. So let's look at this new fingerprint. All right, it says, which do you think is most likely true? It's the same fingerprint of 3 and 10. All right, so they probably came from the same person. If they have the same exact fingerprint, they probably came from the same person. So that's one of the ways people are able to figure out how, if somebody leaves DNA at a crime scene, you can compare that DNA with a bunch of other people's DNA to figure out who left that DNA. All right. So how can you tell whether two DNA fingerprints come from the same individual? So you'd say the pattern of lines on the DNA fingerprint will line up. All right, let's go to page number 12, or sorry, page number 11. So DNA fingerprinting is not the only DNA-based technology. Scientists sometimes look at all of a person's chromosomes in an image called a karyotype. A karyotype shows some types of diseases or disorders, as well as a person's biological sex. So what is a karyotype? It's an image that shows a person's chromosomes. An image that shows a person's chromosomes. All right. And this question says, what karyotype would be what would the karyotype be most useful for diagnosing? Would it be a disease that comes from an error in one specific portion of the DNA, or a disorder that causes chromosomes to separate incorrectly? Well, that would probably be B, right? Because this picture just shows us the chromosomes. It doesn't really show you the individual segments of DNA. But this is important though. You can tell, like I said, you can tell a person's sex from it. And then there are some disorders that actually you can tell if a person has just by looking at their chromosomes. They might be missing a chromosome, they might have an extra chromosome, and a karyotype would show you that. So this last question here on the study guide says, which sex would each of the following combinations of sex chromosomes indicate? So XX would be, right, female. XY would be male. So let's keep going to page 12. So a karyotype shows the X and Y chromosomes. The X and Y chromosomes can be seen on a karyotype. The X chromosome is much larger than the Y chromosome. The combination of sex chromosomes determines a person's biological sex. So we see this little quiz right here. You can go through the quiz, and it will show you if you have the right or wrong answer. So try that out. You can pause my video and you can hit play when you're finished with that quiz, and we will go to page number 13. So 13 is a review. You can go through and check your understanding and go through these questions and submit them. Go to page, you can pause my video and do that. When you come back, we'll be on page number 14. So you've learned that DNA fingerprinting can be used to identify someone who has committed a crime or free somebody who hasn't, to determine someone's family heritage, or to help give patients a more precise medical treatment. In the DNA fingerprinting lab, you will get a chance to actually examine DNA fingerprints and look for matches. I don't think that we actually have assigned you the DNA fingerprinting lab, so that's something you shouldn't really need to worry about. I don't believe. I'll check on that, but I don't think that you need to do that. So that would be it. So there is all your answers for the study guide for 7.4.1. I hope that that helps you out. And I hope you have a great day.